Please welcome to the stage AFTD's Director of Research and Grants, Deborah Niehoff.
Good morning, everyone. As you just heard, I'm Dr. Deb Niehoff. I am the Director of Research and Grants at AFTD. Neuroimaging technologies such as magnetic resonance imaging, or MRI, and positron emission tomography, or PET, provide crucial visual information about changes in the brain to both clinicians and researchers. Today, we're pleased to welcome two experts from leading FTD clinical and research centers to explain neuroimaging's uses and limitations for diagnosing, understanding, and tracking FTD. Joining me are Dr. Joseph Masdeu and Dr. Belen Pascual. Dr. Masdeu is the director of the NANS National Alzheimer's Center at Houston Methodist Hospital and is also a professor of neurology at Cornell University. Dr. Pascual also works at Houston Methodist, where she is the director of the Frontotemporal Degeneration Unit and the co-director of the Neuroimaging Lab. And she is also an associate professor in neuropsychology at Cornell. We're welcome you now to the stage, and thank you for being with us.
Thank you very much. Thank you, Dr. Deb, and thanks to the amazing team of AFTD that has organized everything that we've been involved in. I'm very impressed with the work you work. So, what I'm going to be telling you about a bit is how we can use the tools of neuroimaging to see what's going on in the brain. And one of the tools that you are familiar with is called MRI. So with MRI, we can do a lot of things. We can look at the volume of different regions of the brain, right? We can see also the connection among different parts of the brain. Not only in terms of wiring, how the wires go, but also how the function of the brain is distributed in these diseases. We can also look at blood flow, and blood flow gives us a sense for metabolism, for how active different regions of the brain are. So very useful. But in recent years, positron emission tomography has become an even, I would say, more useful tool. We use both of them combined. But what does positron emission tomography do? What it does is gives us a picture of a particular molecule in the brain. How, whatever it is, glucose, for instance, how is it distributed in the brain of a person? How is this helpful? Well, it is helpful because when somebody has frontotemporal lobar degeneration, the frontal lobe and the anterior part of the temporal lobe are not working properly, and we can see that. And as you can see on the little diagram to the right, that is completely different from what happens, for instance, in Alzheimer's disease, right? You see the blue in those pictures. The blue is the area that is not working well. And anyone in this room, right? If you look at those two pictures, do they look the same? Not at all, right? Any one of you could, looking at a scan, tell that the person that you're dealing with has Alzheimer's or frontotemporal lobar degeneration. Well, this sounds very simple. It's a little bit more complicated, all right? We are not going to be able to make diagnosis just from pictures, but you can see how the pictures, these tools helps us tremendously. But positron emission tomography not only gives us the part of the brain that is affected, but also other proteins that are important in degeneration, like beta amyloid, very important in Alzheimer's. Tau is very important in frontotemporal lobar degeneration. And finally, inflammation is important in both. So we use them to know what the diagnosis is, but also to follow the process so that we can explore what we want most than anything else, what it is that we physicians want more than anything else. We want to find cures for these diseases. We want to find approaches that help us prevent them in families. So that's what we are looking for, and for that, you need a marker of neurodegeneration, something that will tell us whether the medicine is helping or not. And these tools are extremely helpful because they tell us what happens over time in somebody who, of course, is alive, still has the disease. So let me tell you briefly how the scans tell us what part of the brain is affected. I already told you, but these images will help you understand that even better. So you see a healthy control on the left. There is an MRI with a superimposed positron emission tomogram, which is the one that has the bright colors. And you can see that the area of the brain that is affected is very different in Alzheimer's and, for instance, the behavioral variant of FTD, or agrammatic primary progressive aphasia, which is another variant. And these tools have allowed us to differentiate and understand much better the different variants of frontotemporal lobar degeneration, behavioral variant, the non-fluent primary progressive aphasia, the semantic variant of primary progressive aphasia, and then progressive paranuclear palsy and corticobasal syndrome. Now, let me tell you a little bit about a particular variant, which is agrammatic primary progressive aphasia. The part of the brain that is affected is the area that allows us to speak fluently. The person who has this problem has difficulty coming up with the word that he or she wants to say. Well, the part of the brain that is damaged first is the one that you see at the top. Now, you see that behind it, the brain looks brighter. The brain is normal. The area that is affected is bluish because there is little activity, right? But then you continue looking at the brain and you notice that there is an area at the bottom of the brain, quite distant from the one at the top, which is also damaged. So how has the damage spread? Now, this question, you may say, well, so what, right? But this question may lead us to understand why the disease gets worse, right? And also, then we can see whether there is any way that we can prevent the progression. And the protein that we find in those two areas is called tau, T-A-U. You may have heard about this protein before. How many people have heard about tau before in this? Okay, very good. So you know what I'm speaking about, right? Well, so one possibility is that the tau protein, the aggregates that we can see with imaging, begin to happen in the area that we see at the top, where the neurons that are, you see there, the neuron that is higher has these aggregates. And then through the connections of that neuron, they get passed on to a healthy neuron. And this process is exactly what we see in a different disease that is called prion disease. It's not the same. Prion diseases are much faster. They have characteristics that are different, but some of the mechanisms could be similar. So we set out to define whether this is what happens. And basically, the idea is that the area at the top is connected through a bundle of nerves that you see here in yellow to the area at the bottom. That the yellow bundle is called the arcuate fasciculus. And we postulate that tau is going to move from there to the bottom, right? So I'm giving you this example because it tells you how we can use these techniques not only to diagnose, but also to understand what is going on with the brain, what is the biology of these diseases, because from the biology, we will go to the cure, to the treatment. So just to let you know about something that is well known, is that those two areas that I show you are part of what we call the syntactic network. And those two areas are made up in such a way that they allow us to put string sentences together with all the little connectors that we use in language, right? Articles and so on. Well, they are strictly on the left side of the brain. They are not on the right. And they are in red. They are in those pictures that I'm showing you there. And you have very clearly an anterior node and a posterior node. This is from a review. It's not our work. It's a review from the Cambridge linguistics team. But the anterior node happens to be affected in the disease that I mentioned before, which is non-fluent variant primary progressive aphasia. And you can see a nerve cell there from a diagram that Dr. Pascual put together. And this anterior cell, the one that is in yellow there, in green, that connects to the posterior neuron that is in the inferior part of the brain, what we call the temporal lobe. So, and you see the arcuate fasciculus, right, joining the two. Well, if tau moves from there to there, then you may see these changes in patients who have this problem, right? And they have a very clear and characteristic difficulty with language, as I told you before. There's ability to handle syntax and grammar, and fluency are impaired. Well, if it is true that tau propagates from the nerve cell in the frontal lobe at the front to the back, what we are going to see is a stage A, where you see the nerve cell in the front that contains tau already. Now, that nerve cell is still going to be working. And then that nerve cell in the front is sending tau protein, the bad protein, a protein that is folded in the wrong way, to the node in the lower part of the brain, in the temporal lobe. That would be stage one. At this point, the person is not going to come to see us because it's working fine. I mean, all the nerve cells are alive, are still alive. But eventually, as time goes on, it's going to go into a stage B. At that stage, the cell that began to be malfunctioning, the one that had tau at the beginning, is going to die, unfortunately. And then the person is going to begin to have symptoms, right? But what we are going to see also is that some of the fibers that connect the two nodes are going to be missing, right? Because those fibers come from the neurons, from the nerve cells that are in the anterior node. All right, so that is the hypothesis then, that we're going to see tau in the anterior node, tau in the posterior node, and the fibers that join the two nodes are going to be reduced. Well, we can measure tau with positron emission tomography. And when we compare a group of people with non-fluent primary progressive aphasia to healthy controls, we saw the picture that you see there. So as you can tell, there is a difference, but the difference is not on the right side of the brain. It's on the left side of the brain. And you remember that those two nodes are on the left side of the brain, right? So exactly the two nodes contain tau, which is a very strong argument for the fact that tau propagates from the anterior node to the posterior node. Not only that, but when we look at someone with this problem and we displayed the arcuate fasciculus, the fiber bundle. You remember in yellow there, going from the front to the back. You can see how the bundle is thinner. Now in this picture, I have it in green. It's the green bundle that you see there. And if you compare the ones on the two hemispheres, and this is a patient again, what do you see there? And pictures, you have to look at them. So what I see is that the one on the left is a little bit thinner than the one on the right, which is surprising because in the human being, the one on the left is typically thicker than the one on the right. Why? Because our left hemisphere handles language. And that's why the two are not the same. The one on the left side is typically thicker. Moreover, if you notice, it's not only that it's thinner, but it's thinner more on the anterior aspect of the brain. So the left hemisphere, the anterior part is going to the left side, right? So if you look at the left side of that green fascicle, the arcuate fasciculus on that picture, and you compare that side with the right side on the right hemisphere, which is in the on the right side of the picture, the difference is even greater than in the posterior part of the fascicle. So that tells us that there are fibers missing there, and there are fibers missing that are part of the anterior node. The anterior node nerve cells have died. And we can confirm that looking at quantitation with MRI. So you see how these tools allows us also to get into the depth of the changes that are taking place in the brain. Well, let me tell you a few words about another important component of the frontotemporal lobar degeneration disease and progression, and that is inflammation. We call it neuroinflammation, inflammation in the brain. And we see this very clearly when somebody passes away from frontotemporal lobar degeneration and we look at the brain, we see a lot of inflammatory cells. Now, it's too late, obviously, when somebody has passed away to do anything. But also, neuropathology is very cumbersome because you can look at very tiny areas of the brain. You don't get a big picture of the brain. You just look at a very, very small area. And that's why, of course, neuropathology is important, it's useful, but it's not the only technique or the only way of going about understanding a particular feature of the disease. So we use positron emission tomography to gauge what's going on with neuroinflammation. And what we see there is that there is uptake that corresponds very well with the type of the behavior of the patient. And I'll show you some examples in a minute. So, and here they are. So you see, for instance, this lady who had the behavioral variant. She was 50 years of age when we saw her for the first time. And as you know, well, let me explain to you what I'm showing you here. At the top row, I'm showing you an amyloid scan. Amyloid is positive in Alzheimer's, is negative in frontotemporal lobar degeneration. So right there, we have a technique that is very helpful to tell the two diseases apart, right? So hers was negative. The next row is tau. And in her case, there was a little signal there in the frontal lobe that you can see, very, very small, to such an extent that it was inconclusive, and with the more of the signal that you see on the last brain that we show on the right side of that row, that is not brain actually. So that scan was considered also negative. Now, this lady was working when we saw her, but was beginning to have a great deal of difficulty with her work. She requested disability, temporary disability, and she was denied because you know what happens. I mean, if you have any relative with behavioral variant, there is nothing obvious. The non-fluent variant, the person has this problem with language. Behavioral is much more subtle. And that's exactly what happened with this woman. The employer didn't believe that she had anything, basically. So when we did the inflammation scan, we were able to see that there was inflammation exactly in the frontal lobe and in the anterior part of the temporal lobe. You can see that very well. You see those arrows that I have in there. So it was positive. And frankly, this was a very strong argument for this woman to be allowed to take disability. So in this case, it's not only that it was helpful to diagnose, but also help her with her work. Frontotemporal, the non-fluent variant rather of PPA, we have spoken about it before, and you can see how inflammation happens in exactly the same regions that are affected by this disease. Finally, on semantic variant, I'm going to speak about this a little bit more because Dr. Pascual led a study that has had a real interesting outcome that I'm going to tell you about. Semantic variant is unusual. It's not very common, but it has presents in a very characteristic way. So the person begins to have difficulty naming objects and speaking fluently. So we'll have to look for words. The fluency is okay. There is no disfluency, but understanding and sometimes picking up exactly the word that is appropriate, that is difficult. Now, it's most often associated not with tau, but with a different protein that is called TDP-43, and affects the tip of the temporal lobes, and when it is mostly language that is the problem, is the left side that is affected. So here you see this is a diagram, or this is rather a comparison between patients and healthy controls, and you can see how the tip of the left temporal lobe is affected. Now, what is very important is that when we look at inflammation, and I'm not going to have time to explain this picture in a very detailed way to you, but there is, if you look at the very top left picture, you can see how the tip of the left temporal lobe has inflammation in the cortex. And when we compare the place of the brain that has been affected first with inflammation, we found out that inflammation peaks not at the tip of the temporal lobe, but in the area that borders the sick brain with the healthy brain. And of course, what that did is gave us the idea that the process it looks very similar to what goes on in a forest fire, on a brush fire, right? So you have the place where the fire began, is already burnt up. The flames are in the margin. So inflammation does a little bit the same in semantic varia from temporal dementia. This is extremely important because it will guide a type of therapy that Dr. Pascual will tell you in a minute about. All right, so what I did is I told you how the anatomic pattern, how the distribution of the changes that we see with these different traces allows us to distinguish these diseases quite well. And inflammation shows that our inflammation imaging, rather, shows that there is inflammation that could be very, very relevant to the progression of the disease. So with this, I'm going to give the podium to Dr. Pascual, and thank you very much. We are going to entertain questions in a minute.
Okay, excuse me. And before Dr. Pascual gets started, I should have mentioned we will have some time for questions. So if you have a question as you're listening to these presentations, put them on the white cards and raise your hand. Someone will be around to collect them. Or if you are watching virtually, you can put your questions in the chat and we'll be happy to answer them.
Okay, so thank you so much to the AFTD for this invitation to organizing this wonderful meeting with all of us. We are here, all of us, we are a team. We are here to try to cure these diseases. And this is the most important thing that we have to do. And we have to work together. Researchers, families, patients, we are the same team, you know, the same team, the same weight for all of us. So anyway, I want to talk a little bit about treatments, about a little bit of hope. And there are, I don't know if you hear about the all-FTD study that is a big effort, NIH effort for, around the whole country to put all of us together, the experts, the families, the patients, you know, all of us together to be able to treat this disease. And this is very important because in the 5, 7, 8 years, I think that we have been working together, even with the pharmaceutical companies, we were able to bring in the last 5 years, like maybe 10 different clinical trials that before it was, you know, something that we couldn't think about it. And that is hope, because we want to cure these diseases, and we have to do it, all of us together. So I want to talk very fast about some of them. So let me see. So Dr. Masdeu was showing very nicely all of this inflammation, well, this inflammation pattern that we see in frontotemporal lobar degeneration. And we have been seeing that in semantic dementia, but also in other variants that we didn't publish yet, but we will publish very soon. But we know that the next step, I cannot see very well the screens, but anyway, so the next step is to do longitudinal studies that allows us to follow the disease and also to create new clinical trials to maybe to stop that inflammation that could be key or causing the disease, you know. So we are working on that. We have data and we are trying to offer some treatments. Of course, first are clinical trials. But anyway, well, I have here 2 examples because you can see also that in a small group of 10 patients with behavioral variant frontotemporal lobar degeneration, we saw also inflammation, and the same with the non-fluent variant primary progressive aphasia. So inflammation is a key, no? This is, and also, for example, this is a patient with the non-fluent variant of primary progressive aphasia, and you can see the progression in 3 years. So this patient was progressing very slow, but you can see the amount of inflammation in the bottom line of this slide. So we want to do something. So thanks to all of this data and more, for example, for all of you that you are participating in this big observational study that is called all-FTD, you know, we get a lot of blood, CSF, and many other data. So we were able to come with the first treatment for semantic dementia. Well, treatment, it's a clinical trial. We don't know if but at least we have something to offer and something to try together, right? So this clinical trial, what it's trying is to give a medication that is called verdiperstat, and we are trying to reduce inflammation. And we expect that the patients that are going to progress slow or maybe stop the progression, that will be ideal, right? So the duration of this treatment is for 6 months. So pills, no infusion, so it's simple than other clinical trials. So it will be, you know, we will be enrolling in this clinical trial. I think it's 64 patients. The idea, it will be as well, I know it's a little bit late, so I'm not going to stop in the specifications of the clinical trial. But if you are interested to participate, you have a QR there, I think, no? And the five sites of all FTD that are running the clinical trial. UCSF in San Francisco, the PI is Dr. Lubekat, that he's the PI, the general PI of this clinical trial. In Houston Methodist, we are also running this clinical trial. And Dr. Masdeu and me, we are the PIs of this clinical trial, but also you can participate in Mayo Clinic, you can participate in Northwestern University, and also in the University of Pennsylvania. So this is the first one, and we have another first one that is for the non-fluent variant of primary progressive aphasia. So we are trying to modulate, inflammation through the interleukin-2 plus acetabet medication that will be a subcutaneous injection during 6 months, only the first 5 days of each month, to see if we are able to reduce inflammation in this disease. So, as you know, we can see inflammation, but also in the blood we saw, and you can see the bar that is lower, that is the non-fluent variant of primary progressive aphasia data, that the regulatory T lymphocyte activity is very low. So with this medication, we want to increase the activity of the lymphocytes. So this is the mechanism of this clinical trial. So right now, what we are going to do is to do PET scans first and all type of study, all the inflammation in blood and CSF and neurodegeneration. Then it will come the treatment, and then we will repeat all these measures. So of course we are expecting also being the cognitive status to be stable or even improve a little bit, or, well, we will monitor that. So this is also, as I told you, for 6 months. We have the hope that it could help, and it could be a future treatment for this disease. So right now, the only site for the all FTD site is Houston Methodist, so you can participate here. Dr. Faridar, Dr. Masdeu, Dr. Appel, and myself, we are working in this clinical trial. Right now it's small. We will be enrolling 12 patients, but at the end of the year, we are going to do a bigger trial with around 60 patients. So this is a hope. And I want to finish because we would like to listen to your questions, but I would recommend you to participate in this study, because really, again, I was telling you at the very beginning, we are the same team, we are working together, we want to keep working together to try to find a cure for these diseases. And I finish with that, and we are ready for all the questions that you have for us.
Thank you.
I'm sorry for running a little bit with this.
Thank you, and thank you particularly for that exciting information about trials that are in progress and trials that are on the horizon. If people here think they might be interested in participating, how would they go about doing that and finding out more information?
Well, the QR that was there, it has all the information. I think I'm leaving some flyers around with the information of these clinical trials, also the all FTD study, in case that is new for some people. But you will have the information. There are my team from the Frontotemporal lobar degeneration that is in the bottom. They have information also, Victoria Arboni, Sarika, Kaylee, Dr. Masdeu, myself. We can give you information about all of these trials.
Yeah,
Plus, there are many pharmaceutical companies here. They have wonderful trials also, for example, for the GRM mutations, so in the last years, really, and I think it's thanks to that study, big study, that we are running, and we want to go as fast as we can. But we have to work, all of us together.
Do we have any questions from the audience? And have you given a card to, nope. Then I think you can go ahead and ask your question.
Hi, thank you so much for this information. It's been helpful. As a care partner, I'm familiar with PET scans and MRIs, but I wanted to understand a little bit more about SPECT imaging.
Yeah.
There's been some controversy around that.
Yeah.
Very good. Yeah.
Explain, and
Yeah.
it doesn't have particular relevance to certain variations.
Exactly. Very good question. And the question is whether SPECT, which is single photon emission computed tomography SPECT, is also helpful in addition to PET for the diagnosis of frontotemporal lobar degeneration. And the answer is yes, absolutely. The study with SPECT takes longer. It is easier for the person to move. So you have to be very careful choosing the place where the study is done, because otherwise you may get a study that is not that helpful. But if it is done properly, it is very helpful. So I didn't put it on my slide because we had only 45 minutes for this session. My apologies.
Can you just elaborate? Helpful in what way?
Yeah, it is helpful because, do you remember I showed you that PET shows that the front of the brain is affected in frontotemporal lobar degeneration? You can see the same thing with SPECT. Absolutely the same thing, yeah.
And I think we do have some other questions now from the audience.
We do have a question from someone in the audience on one of the cards that they handed in, and it really relates to something that will be covered partially later today at the 3:45 session, but maybe our speakers could respond to inflammation of the brain. What would they like to share with us about lifestyle factors and what they would say about diet or other issues that could in some way possibly moderate or impact inflammation? Would you like to share any thoughts on that?
Yes, of course. You know, we are beginning to understand how some behaviors and food modifies the inflammation in our bodies. There are some substances or some foods that are cause more inflammatory changes, but having dealt with inflammation a bit, I must tell you that the propensity of a person to have inflammation is highly dependent on their makeup, on their genotype. When somebody begins to have frontotemporal lobar degeneration, and you know, you think, well, is there anything that I could do to modify the course of the disease? I doubt that modifying inflammation is going to do anything. In the Alzheimer's world, already we know that giving anti-inflammatory drugs to people who already have symptoms, basically, unfortunately, does nothing. And so I wouldn't bother your loved one with a diet that is very constrictive. I mean, I know there are a lot of people out there, you know, giving information on diets that are going to improve the process and so on. I'm a little bit skeptical about that. I mean, once we have a clinical trial that shows us that that works, then I will advise you to perhaps make your loved one lives a little bit less pleasant, because they won't be able to take this or that or the other thing. But at this point, I frankly cannot do that. We do have a study going on, the Pointer study, looking at these lifestyle issues for Alzheimer's, which is, you know, the population that have Alzheimer's is you know, much larger. Speaking about lifestyle, I would like to share with you something in 1 minute, which is that I'm a neurologist who has been taking care of people with dementing processes since I was at the Albert Einstein College of Medicine in New York quite a few years ago. And you know, one thing that you should never do is put yourselves in the shoes of the person with frontotemporal lobar degeneration, because the way the disease is experienced by the person is very, very, very different from the way we experience it. You know, at the National Alzheimer's Center here in Houston, we have a questionnaire for depression that is universally used, the Savage scale. And we ask the the people who fill out these questionnaires to make sure that that particular questionnaire is filled out by the patient, not by anyone else. Well, you know, when I get a Jesabali questionnaire with a very high score suggesting depression, oftentimes I look at the relatives and I ask them, who filled out this questionnaire? Well, you know, so-and-so just didn't want to fill it out or whatever, we filled it out, right? So, you know, the perception of the patients, and you probably have, those of you who have relatives with this disease probably know that very well. I mean, their perception of the disease is much less grim than the perception of the person who is looking from outside in. So I think that's very important because, you know, even in the course of the disease, those years, you can still enjoy your relative, I mean, you know, have a good time together, and know that they are not suffering nearly as much as you are. This is a disease that really makes the family suffer more than the patient suffers. We have a wonderful way in our brain to compensate for these horrendous diseases, and it makes sense, right? If I have a problem with my finger, I can objectivize it. This is my finger that is hurting. When the problem is in the brain, the person cannot objectivize it, and that is wonderful. I mean, obviously this is a horrible disease, but I think knowing that you know, if basically the family is happy, the patient is going to be happier. Maybe, particularly at the beginning, they are not happy. They can be very distressed by what's going on, but you know, particularly as the disease moves on, they can be quite you know, comfortable so long as they are well taken care of and things are appropriate. So just as this little message, because I think it's so important to lessen the distress caused by this terrible disease.
Thank you. And I think we have time for one or two more questions.
So actually, we are going to pivot, and I just want to acknowledge that there were quite a few questions, both virtually and in person, about the research study that you spoke about. And I'm sure that there was information on your slides about where people can get more information in terms of genetics, not genetics, a prime candidate. But there was a lot of interest, and we'll definitely be sending people your way. So thank you very much for including that in your presentation.
Mm-hmm. And thank you both for being here. Thank you for answering questions. And thank you for your questions, those in the audience.
Thank you so much.